Hello everyone and welcome to my tech video introduction and seminar report. My name is Sari and currently I'm working in lab 510 of Professor Xiao, and my currently work is focused in the wound healing process of the Zebrafish model. And this will be the content for my video today. So first I will talk about my tech video, and for the paper report we'll be including with introduction, material, method, result, and conclusions. For the beginning of the video, I will talk about my TED video first. So this is the video I choose because currently I'm working on the study about the zebrafish in Professor Xiao lab. So I'm choosing this video, name title is the Zebrafish. Practically people, how to save human species. The speaker is Dr. Wendy Leonard. So this will be the link of the video and the video right here. But and to conclusion that video for you, we will solve a big question like, did you know that zebrafish can be the salvation of the human species? Why? Because of that. The zebrafish in biomedical research, they share 70% of the human genes and 84% of the genes like to human genetic disease. Their immune system and drug response are similar to human, making them valuable for modeling disease like cancer, diabetes, and heart disease. They reproduce rapidly and cost little money, like cost 6.5 cents per day per tank, and have transparent eggs for easy observation. Some of the unique factors of the zebrafish, like they can generate the organ like spine, heart, and retina. It's useful for study brain disease and depressions and autism. And because of that, we have some very useful application like environmental toxin impact study, drug efficiency, and addiction research, and can bring some potential breakthrough in regenerative medicines and tissue engineering. So this will be the paper I choose because it relates to my study in the lab. So the title will be the live imaging of the cutaneous wound healing after rotary tool injury in zebrafish, and this was published in the Journal of Investigative Dermatology, and Q1 ranking and impact factor is 7.59. For the introduction of the study, the cutaneous wound healing is a complex process requiring the coordination of different cell types to ensure the wound closure and tissue repair, and it is characterized by 4 overlapping phases. First, the homeostatic, in which the blood clot is formed. Second, the inflammation, where the immune cells are recruited to the site of the injury. Third, the proliferations, including the skin re-epithelialization, angiogenesis, and fibrogenesis. And the last is remodeling during the vessel operation and collagen remodel, usually resulting in the scar formation. So the poor healing is common in elderly and those with diabetes, leading that the chronic wound susceptible to infection, according to previous study. And understanding how healing is altered in aging and disease is the critical to generating better therapy to ameliorate defecting healing. For understanding and studying the wound healing process, the study used the zebrafish as a model. So the zebrafish will provide an attractive vertebrate model for studying tissue injury and repair in vivo, with their high regenerative capacity and conservation of major state of cutaneous wound healing. So unlike the mammal, which have like a huge important insight into the cell type and signal-driven healing, but unlike them, the zebrafish have optical clear embryos and larvae, and pigment-free adult zebrafish lying are available, facilitating high-resolution optical imaging of cellular and even the subcellular feature in insect animal. So in addition, the author have developed a method for long-term optical imaging of intubate living adult fish. These features are complemented by a plethora of transgenic and mutant fish line available for the direct visualizations and functional manipulation of the different epithelial, immune, vascular, and other cell population contribute to the wound healing. For material and method, here we got some image about the rotary tool used to create the cutaneous wounding. So for example, we have here is the overview image of the apparatus used for generating the cutaneous abrasion injury. And for B, C, D, E is some detail for the tool to cause the wound. And F is the holding platform. G is like a focus image of the operating table for fish. And H, Y, K is the image of the mounting of the fish onto the holding platform, showing the platform with the wet filter edge. A fish play on the platform on the top of the paper, and mount fish with immobilized on the platform using the anchoring rod. So they're using platform and using the rod to immobilize the fish during the operation. So after we operate the wound on the fish, we have to observe the wound healing process using the body P standing. So first we gotta prepare the dye first. So the body P forehead fluorescence dye reconstitute in 1 ml of DNSO to make the 5 mg/ml stock solutions. And for the process, it's very easy. We just need to adult fish to bath in the 20 nanogram/ml of PodiPie solution, which means we use 2 microliter of PodiPie stock solution and dilute in the 50 ml of system water and bath them for at least 2 hours. So after the bath, the fish were rinsed and for 1 hour in clean system before imaging. So the author get the protocol represent from the previous study in from Lumakin in 2021. And after that we do the image acquisition and image processing and analysis. So this will be the method to showing the preparation and generating of the cutaneous wound in the zebrafish using the rotary tool. So before we do the operating, we select the adult fish from 3 to 13 months old, and we unconscious the fish using the Trican solutions. So after they unconscious, here we got the video for us audience to fully understanding the process. The author have kindly provide us the video where they preparation and generating of the cutaneous wound healing of the wound. So first, before injury, you can see the fishes can swim very healthy, which means that they select the fish with the good conditions for studying. And after that, addition of the filter paper with the Trican water to holding platform, and we loading the fish onto the holding platform. You must be very careful because you don't want the fish to have any else injured. So after that, we secure the fish into the platform using the anchoring rod. And the rotating insert to the angle fish. And lining up the rotary tool to the fish to the side of the injury. And after that, we create the injury with the rotary tool for 10 seconds. And result, we've got the cutaneous injury like we designed, and after the injury, the fish still can swimming very healthy, which means that the method does not cause the death of the fish. So for the result, here we got the image of male and female fish before and after cutaneous wounding, showing the consistent wound formation using the rotary tool. Because when you study, you don't want the wound to be you want it to be consistent. So the qualification of the wound area which is thought representing a single fish. Here we got 13 fish, and arrow bar indicate the mean area of the standard deviations. So the rotary tool is turned on the speed 3 to operate, which means it's 8,500 RPM, and press gently into the side of the fish for 10 seconds. So when we operate the fish, the author operating the fish on one side of the fish for every fish, and that will creating a 1/16 diameter abrasion incher that approximately 2.4 106 micrometers square in area. So for understanding the process of the wound healing, so first we got the time-lapse imaging of the skin re-epithelializations after wounding. So here we got the still and time-lapse image of the bee acting of fish before and after cutaneous wounding. So first we got the still image of the fish prior to the injury, including the zoom out image and close-up image of the wound site. So time-lapse, this is the time-lapse of the skin re-epithelialization from 0.5 to 17.6 hours after injury. And after that, we got the high magnification time-lapse 18 hours after injury, showing the infiltration of many cell types into the wound area. And here, to TBI wound close, we got the still image of the wound area 2 days after injury, showing the wound had been closed. We got the figure of the reproducible cutaneous wound can be made with the Rotary tool. With the figure C right here is a magnified image of the flank of male on top and female fish on the bottom before and after cutaneous injury after 20 days post injury. And D is a confocal image of the flanks of the bee acting transgenic of the male and female. The same, the male is on top and female is on bottom. After 20 days, after and before and after 20 days of injury. So the scale bar is for image C is 2000 micrometers and image D is 1000 micrometers. So we collect the stereoscope image of the male and female fish for 20 days after injury. So we can observe here that the private blood clotting occurred immediately after post-injury, and the wound re-epithelialized by 1 day post-injury, or DPI for short, and we call it DPI for short, with the impingement area shrinking over 20 days. Confocal time series of injury of the bee-acting transgenic adult fish show that the bee-acting expressed the cell in green, and before injury, the GFP is mostly in scale, and GFP lost in wound area immediately after injury. By 1 dBi, the GFP pre-expressed throughout the wound site, indicating the seal wound. And time-lapse confocal imaging from 0.5 to 17.6 hours post-injury. So epithelial cells moving into the wound site of immediately post-injury, confirming the rapid wound closure. Like I mentioned before, the wound healing involves 4 phases: hemostatic, inflammation, proliferation, and remodeling. So the neutrophil are crucial during the inflammation phases. The neutrophil recruitment is a crucial early step in the wound healing process. These immune cells are among the first responders to the wound, drawn by the chemical signal from the damaged tissue. Upon arrival, neutrophils help prevent infection by engulfing pathogens and debris, releasing the antimicrobial substance and recruiting other immune cells. They are timely removable by macrophages, then pave the way for tissue repair and healing. Proper neutrophil functions is essential for effective wound healing, while the circulations can lead to chronic wound or delayed recovery. So here we got the video about the neutrophil recruitment after the cutaneous wounding. So the still and time-lapse image of the, I'm gonna call this DS Red 2, fish stain with the polyp before and after cutaneous wounding. So the schematic of fish show approximate site of the wounding. So here we got the still image of the wound area before injury. Show the scale intake and neutrophil evenly scattered throughout. And here we got the time-lapse video of the neutrophil recruitment from 0.5 to 17.25 hours after injury. So like in the video, you can see a mass influx of neutrophil is seen during the indicate times, suggesting that neutrophil is respond quickly very quickly to the cutaneous injury. Body labeling skin re-epithelializations occurring simultaneously with the neutrophil recruitment. To understanding the wound healing process, we also need to study the neutrophil behavior. So here we got a movie about the neutrophil behavior after the cutaneous wounding. Still and time-lapse image of the DS-Red2 fish before and after cutaneous wounding, showing the neutrophils in magenta and scale in blue. So here we got the still image of the wound area before injury, show intact tissue with neutrophils scattered throughout. And a still image of the wound soon after injury, including a zoom out and close-up image of the wound site. So here we got the zoom in time lapse of neutrophil behavior from 0.5 to 17.5 hours after injury, showing the individual cell in the left movie and tract cell containing spot, magenta color you can see, and with the trigon cell in cyan color. Despite the last 15 minutes of the movement in the right movie. So movie are paused at the following time to indicate the specific neutrophil behavior. At 30 minutes, you will see the initial migration. For 20 minutes, you will see the amplified recruitment. 28 minutes, you will see the neutrophil clustering. And 35 minutes is the wound infiltration. So this agrees with the previous report that's showing that unlike in the mammal, where re-epithelializations occur in the later phase of the wound healing, the zebrafish as a model showing that the re-epithelialize their wound as one of the first steps of the repair process, according to the report of the Richardson in 2013. And after initial wound closure, scale can be seen gradually regrowing over the injury site, covering the entire wound by 10 days after post-injury. And to examine whether some of these might be neutrophil recruit into the injury site, we collect some high-resolution confocal image of the DS-Red transgenic Casper mutant pigment deficient adult fish as a continuous time-lapse from 0.5 to 17.25 hour post-injury, and then at interval there until 15 days post-injury. So although the neutrophils are presented to the surface of the fish prior to the injury, there was a rapid influx of a large number of neutrophils into the wound site after injury. So there was already extensive migration of neutrophils to the periphery of the wound after 2 hours post-injury, with neutrophil recruitment peaking at 1 day post-injury. And this initial burst of neutrophil recruitment was followed by more gradual decline in neutrophil localizations to the wound site, and with neutrophil level close to the baseline by 15 days post-injury. And all of this which suggests that zebrafish have an acute neutrophil response to the cutaneous abrasions wound that dissipate quickly. One more information very important to study in the wound healing is the macrophage movement. So after the cutaneous wound, the macrophage play a pivotal role in the healing process. They are recruited to the wound site through the chemotactic signal and differentiate from monocyte into macrophage. Once at the wound site, they clear debris and pathogens, regulate inflammation, and promote tissue repair by transitioning from a broad inflammatory state to a broad healing state. So the macrophage also stimulate the new blood vessel formation and extracellular matrix remodeling. So that is very essential for tissue regeneration and wound closure. So proper macrophage function is crucial for efficient healing, while deregulations can lead to chronic wound and delayed recovery. So we got the movie here is a macrophage movement after the cutaneous wounding. So the still and time-lapse image of the GFB fish before and after cutaneous wounding showing the macrophage in green. So here we got the still image of the wound area before injury show the macrophage completely covering the skin. And here we got the time-lapse video of the macrophage behavior from 0.5 to 16.5 hours after injury. Show that macrophage streaming into the wound site after injury. And here we got the high magnificent time lapse, 16.5 hours after injury. Here we got the figure representing the revascularizations of the cutaneous wound. We got figure A here is the whole fish overview. B and C is a magnified flank confocal image. With B in green is a lymphatic vessel and C in magenta is a blood vessel. So figure A here we got the white dot box indicate the approximate site of injury for figure E to B. And for figure D is the qualification of the percent of the wound area revascularized by the blood vessel or lymphatic vessel at the time point following continuous injury. So angiogenesis is the formation of the new vessel. It's crucial for wound healing as it ensures proper blood flow and drainage of the fluid in the healing tissues. In this study on adult zebrafish, the researcher examined how blood and lymphatic vessel regrow after the skin injury. They use a double transgenic Casper mutant zebrafish to track this change over 15 days post-injury. You can see in figure D. And some key findings in this figure we can see is in the normal vessel pattern. For lymphatic vessel, it's in uninjured fish, lymphatic vessel forming a clear pattern with main lymphatic vessel run along with the midline of the fish with smaller vessel branching out. And for the blood vessel, the skin blood vessel have an irregular and branching pattern, while the muscle blood vessel are thinner and run along the muscle. And for the post-injury vessel growth, we have like the first phase is the initial phase from day 0 to day 4 after injury. So right after the injury, the wound have fragments of the several blood and lymphatic vessel. But by day 2 post-injury, these fragments start to disappear and new vessel growth begin to rapidly. And by day 4, more than 75% of the wound area is revascularized. The second phase is the later phase, from day 4 to day 10 after the injury. So after day 4, the growth rate slow down, but the vessel continue to grow into the wound area. And by day 10, around 90% of the wound area is covered by the new blood and lymphatic vessels. So after that, we get the continued remodeling. So even after 10 days, the vessel growth and remodeling continue for several months, adjusting and strengthening the new vascular network. For summaries, the studies show that the zebrafish skin wound healing efficiently through the rapid initial growth of blood and lymphatic vessel, followed by the slower growth and ongoing remodeling. And this process ensures that the healing tissue receive the adequate blood flow and fluid drainage. So after some detailed study in the wound healing process, we can go with some conclusion in this study. So the zebrafish inhibit the rapid wound closure, with reepithelializations occurring as one of the initial steps, unlike the mammals model. So the live imaging technique revealed the dynamic change in the cell behavior during the wound healing process. Zebrafish display an acute neutrophil response to the cutaneous abrasion wound, with the rapid influx of the neutrophils followed by the gradual decline. And angiogenesis play a crucial role with the rapid revascularizations of the wound followed by the vascular remodeling over time. And zebrafish serve as a powerful model for studying the coordination of the cellular and molecular process involved in the cutaneous wound healings. So based on this study, I will have a lot of background for my personal study because I'm also studying in the zebrafish wound healings. Thank you so much for listening to my presentations, and if you have any question, please comment down below. I will try to answer to all of you. Thank you so much.